Dr. Ravigadevi, who will give her talk on oil palm phenolics from bioburden to bioactives. Over to you, Dr. Ravi. Thank you.
Thank you very much, Madam Chair. A very good afternoon, ladies and gentlemen. I have the unenvious task of having to give this talk at 2 o'clock, and I promise I'll try to keep you from dozing off. Okay? First of all, I'd like to thank Dr. Sundram for inviting me to give an overview of the oil palm phenolics program. And as Dr. Sundram mentioned yesterday in the opening, this is the new kid on the block. And I will give you the background and paint a very wide or broad stroke of the research activities. And my collaborators will then fill you in on more of the specifics. Okay? Now, my collaborators, the team members, of course, we have the MPOB team. The original team was comprised Dr. Tan Yu Ai, Dr. Sundram, who was then in MPOB, and myself. And we got this idea actually during a coffee break, which just goes to show how important it is to relax and have a break. And the team has since expanded in MPOB. And we have also broadened our horizons, and we collaborate with MIT. And the MIT collaboration is led by Professor Cho Kyun Ra, who has been described as insanely committed to the phenolics program. And this afternoon, Professor Sinsky will talk about our collaborative efforts. He is the sanely committed one. And the others in the group are Dr. Sam and Cassie, who's here. Okay. We're also very pleased to have with us Julia Bolsinger, and she's from Brandeis, and she will tell you about our efforts at looking at anti-diabetic properties, and this collaboration is led by Professor Hayes. Mahinda from CSIRO leads the program at CSIRO on cardioprotective effect of oil palm phenolics. And unfortunately, we are not able to have Dr. Ali, who is from the Brigham Women's Hospital at Harvard. And, but I will give you a flavor of what he has done. And our youngest member of the collaboration is actually Dr. Smiti Gupta from Wayne State, and Pramod is also on this team. Okay. Now, a major target of the oil palm industry is to extract as much oil as possible. And after the oil is extracted, most of the non-oil components or water-soluble components are channeled in the aqueous stream and ultimately end up in the effluent pond, and they're known as palm oil mill effluent. Okay? And in our pursuit for oil, many of us have actually forgotten that the oil palm is actually a fruit, and in fact, a very beautiful fruit, brilliant colors. And these brilliant colors are usually associated with good nutritional properties. If you look at the blackberry, strawberry, well, grapes, they all have got nutritional properties, and this is because of the phenolic components. Mainly anyway, because of the phenolic components. And these phenolic components, being water-soluble, we realized actually are finally winding up in the effluent pond or drain. And it occurred to us that what a waste this is. And the palm oil industry in Malaysia alone generates about 45 million tons of palm oil mill effluent or POM. And this obviously is a bioburden. It has serious environmental implications. But we saw it as an opportunity. 45 million tons of POM to work with, and these are all the water-soluble compounds are going in there. So we did a quick analysis, and we found indeed there are some valuable compounds in the effluent, and we were especially interested in the phenolics. Okay? We developed a very simple process, solvent-free, and we've built a pilot plant at Labu in Negeri Sembilan. And today, whatever experiments I describe will be based on this extract that is produced at the pilot plant at MPOB, because this product is not freely available yet. Okay? Now, there's increasing scientific evidence to show that fruits and vegetables, okay, because they're rich in phenolics, have got many biological activities, and this goes all the way from antioxidant, antimicrobial, anti-ulcer. So we wanted to see where the oil palm phenolics fit into this array of bioactives. And we found that indeed it is rich in many bioactives, and we confirmed this with in vitro, cell culture, whole animal, and microarray studies. I can't describe all the studies, I will just select a few. Now, for in vitro studies, we did several antioxidant assays. One of them was the free radical scavenging activity using DPPH, or diphenylpicryl hydrazine, and this is a stable free radical. And we found that indeed, the oil palm phenolics have got potent free radical scavenging activity. Now, since it has got good antioxidant activity, we realized then that it has perhaps potential to work against oxidative stress. And one of the models we looked at was looking at neurodegenerative disorders. This is because oxidative stress is a major factor in the pathogenesis of neurodegenerative diseases. And the brain is particularly susceptible to oxidative damage because of the high consumption of oxygen and because it has high quantities of polyunsaturated fatty acids. And we knew from the literature that polyphenols have potent antioxidant properties against neurodegenerative disorders. So we looked at cell culture, microglial cell culture. Now, microglia, they are the resident immune cells in the brain, and they play a pivotal role in immune surveillance. Okay? They are a key source of pro-inflammatory factors such as NF-kappa B, TNF-alpha, and inducible nitric oxide synthase. And these are all overexpressed in many neurological conditions. Okay? So in this assay, this was actually done by the MIT team, and they used the microglial cells, and they activated it using lipopolysaccharide. And you can see when it's activated, you have a large amount of INOS, or inducible nitric oxide synthase. But LPS, in the presence of various concentrations of oil palm phenolics, you can see that there is a decrease in this protein. Okay? And you can see it more clearly here. In another experiment, similarly, we activated the microglial cells with hydrogen peroxide, and we measured the NF-kappa B using Western blot again as before. And again, you can see here in the presence of hydrogen peroxide, the microglial cells are activated and produce a lot of NF-kappa B, whereas in the presence of oil palm phenolics, it is significantly reduced. Okay? We also looked at the anti-cancer properties using cell culture, and this was done in Wayne State University by Dr. Smitty. She used 2 pancreatic cell lines, and using 2 different assays, both the MTS assay as well as a clonogenic assay, she showed that oil palm phenolics induce an anti-proliferative effect, and it is dosage dependent. Okay? So this is anti-proliferative. She also showed that oil palm phenolics induce an apoptotic effect on the same 2 cancer lines. And here, this was done using an ELISA kit, and this was done using flow cytometry. Okay? And in our labs, we also showed that the oil palm phenolics have got potent inhibitory effect against HIV reverse transcriptase. Now, some of you may know that currently the 3 main methods of treating HIV is either by using drugs which have inhibition against HIV reverse transcriptase, or protease, or integrase. So we had pretty good inhibition with for the reverse transcriptase. So did we for the protease. I'm not showing it here. And more recently, we looked at the integrase, and we have different fractions. Just to show you, this is OPP. You've got old OPP and new OPP, meaning we had 2 different batches using slightly different processes. And in both cases, we see a very pronounced inhibition of the integrase, okay? Meaning that the oil palm actually at some point might be used. Sorry, not oil palm. The oil palm phenolics might have value, okay, against HIV. Okay? Now, those were all cell culture studies, and they're good, they give an indication, but we really need to move on to animal studies because the physiological response, obviously, is not the same as what you see in cell culture. So, this experiment was looked at to see if oil palm phenolics had any cardiovascular benefits, and this was done by Dr. Mahinder of CSIRO, and he looked at blood pressure, He looked at vascular relaxation as well as the effect of all palm phenolics on tachycardia and fibrillation. And he will talk to you more about this, but basically the bottom line is in every one of these assays, all palm phenolics had a positive effect, showing that it has good cardioprotective properties. In our own labs, and this was done by Anisha, and I think Nagan also was part of this, we used a rabbit to see the effect of all palm phenolics when the rabbits are fed an atherogenic diet. Okay? And after the period of study, the rabbits were sacrificed, the aorta removed, and they were examined. And the aorta, well, usually in atherogenesis, it progresses from lesion-free, which is obviously the best, to fatty streak, followed by fatty plaque, and finally the worst condition is fibrous plaque. So we can see that all palm phenolics had the most lesion-free areas, or in combination with TRF, a tocotrienol-enriched fraction. So these two did the best. And if you looked at fatty streaks, here again, all palm phenolics significantly less fatty streaks, and similarly with in combination with the TRF fraction. Okay? And you can see the effect here. I won't explain. It's really down. And what was interesting was there was absolutely no fibrous plaque when both all palm phenolics and the enriched tocotrienol enriched fraction was given to the rabbits. And just OPP alone, too, significantly reduced fibrous plaques, again showing that it may have good cardioprotective properties. Okay? And seeing is believing. This is the aorta we removed, and you can see that this is control on an atherogenic diet. This is with palm phenolics and vitamin E. You can see it's much clearer. Okay. Next, we also wanted to see the effect of all palm phenolics on diabetes. It's obviously a big problem in the Western world, but also in Malaysia. And so this work is actually done at Brandeis. So I won't steal the thunder from Julia, who will be presenting the details of this work. But I would just like to introduce you to this rat. It's called the Nile rat. It lives along the banks of the River Nile. As long as it lives along the banks of the River Nile and it's burrowing for its food and it eats, well, grain and whatever, it's fine. But if you bring it into the lab and you give it good lab chow and no exercise, it develops what we too develop, which is metabolic syndrome. And especially the male rats develop diabetes and, as I said, qualities or properties of metabolic syndrome. And Julia will give you the details, but basically we noticed that if you fed these Nile rats all palm phenolics, it significantly reduced the glucose level, sugar level, as well as the triglyceride level. So Julia is the next speaker, and she'll elaborate more on these results. Okay? Now, macular degeneration. Now, this work is done in collaboration with Dr. Ali Hafizi. And you may know that age-related macular degeneration is the most common cause of vision loss worldwide in older individuals, that is, individuals over 50 years. And I've tried to show you here what it is like to have macular degeneration, normal vision, and this is vision with macular degeneration. So it is really quite a debilitating condition, and this is another example. Now, vascular endothelial growth factor, or VEGF-A, has been identified as the key molecule that's responsible for age-related macular degeneration. And currently, actually, the only therapy to date is this anti-VEGF treatment, where you actually inject it with anti-VEGF. But not all patients respond to anti-VEGF. It is expensive, and it is painful. So we were interested in seeing if that all palm phenolics could help in any way, could ameliorate the condition. And just to explain to you, how does macular degeneration occur? Basically, it's some form of angiogenesis. So the blood vessels leak and they grow into the subretinal space in a process known as choroidal neovascularization. So you can see here that it's leaked. And yeah, so these are the CNV spots. So in this experiment, mice, okay, the cornea of mice were implanted with VEGF, okay, to induce macular degeneration. And before this, the mice were either fed on water or on all palm phenolics. And you can see that these are the mice, you can quite see it here, that were just on the control. And these are with PFJ, is another name for OPP, all palm phenolics. We either call it OPP, or the Brandeis people and some of the people in the US prefer to call it PFJ, palm fruit juice. Okay? So you can say that there is a significant reduction in angiogenesis in the eyes of these mice that have been implanted with VEGF. In another experiment, choroidal neovascularization was induced by shooting a laser beam into the eyes of the mice and then measuring the level of neovascularization. And here you can see it, but maybe this is clearer. There was a significant inhibition of the choroidal neovascularization. Now currently, well, in addition to VEGF, which is actually the only therapy, but taking extracts of bilberry extract is supposed to help relieve the condition a bit. But I don't have the data here. These results were much better than with bilberry extract. So that's the whole animal studies. And we also wanted to look at the effect on gene expression, and we did this using microarray or a DNA chip. And reason we wanted to do this is actually to try and attempt to understand the molecular mechanisms underlying these physiological responses to OPP. Okay? And in this experiment, this was on mice that were fed an atherogenic diet. Okay? We could see that many genes were upregulated or downregulated. You obviously can't read what the genes are, but the message here is that this is the control. Okay? Yellow is upregulate, blue is downregulate. And when the mice were on an atherogenic diet, there was a significant upregulation of many genes. However, when they were given all palm phenolics, the exact opposite occurred. Okay? There was a downregulation. Similarly here, when there was a downregulation of genes in the control, there was an upregulation in those given all palm phenolics. So what this is showing is that there is actually a modulating effect, and the all palm phenolics are doing the exact opposite of what an atherogenic diet would do to the mice. And in this one, we looked at the anti-cancer effects of all palm phenolics on mice. Earlier, I talked about the cell culture studies that was done by Smithy. This is on the actual mice, and this was done in my laboratory. And the mice were injected with myeloma cells, and indeed, when you inject them directly at the high doses that we did, all the mice actually developed the tumors, skin tumors, but you can see that it was significantly lower. That means the tumor load, the tumor volume was significantly lower in the mice that were fed all palm phenolics. Okay? The tumors were removed, and we looked at the gene expression in the tumors, and what we found was there were some genes that were upregulated, but many genes of the cell cycle were downregulated in the tumors of mice fed all palm phenolics. Another set of genes or group of genes that were downregulated were the striated muscle contraction pathway. In this particular experiment, we did not see much apoptosis, but we saw more cell cycle arrest and downregulation of the striated muscle contraction pathway. And this summarizes actually the data we got for the microarray on the tumors. So basically, in the presence of phenolics, there was a downregulation of the cell cycle network and all palm phenolics, implying that the all palm phenolics probably inhibit tumor growth by inducing a G1 S phase arrest. There was also a downregulation, as I said, in the muscle contraction network, suggesting a reduction in cell division or cytokinesis and cell movement. That's invasion and metastasis. And as I said, surprisingly, we didn't really see many genes involved in apoptosis, and suggesting that at least in this animal model, the phenolics acted as a cytostatic rather than a cytotoxic agent. And it might be related to the model. It might also be related to the concentration tested. But what is important is that all palm phenolics obviously executes different mechanisms in the fight against cancer. Okay? And so that's more for disease conditions. We're talking about heart disease and cancers. But more and more people are now interested in wellness. You know, I want to take something before I fall sick. So we wanted to see what's the effect of all palm phenolics on healthy rats. And in this experiment, mice. The mice were given a all palm phenolic diet or normal diet with water. And we noticed that several hundred genes were either upregulated or downregulated. I shall not go into all these genes, but it's quite interesting to see that the organ that responded the most in the mouse was actually the heart, okay? And the genes that were significantly actually upregulated were the caloric restriction mimetics. What is this? I think many of you would probably know that if you want to live longer, you cannot eat like what they provided us for lunch today. You really have to starve, okay? So the less you eat, the longer you live, which is a bit kind of sad. But what we found was actually the phenolics were actually mimicking as if you were on a diet. So that was quite nice. And it also upregulates fatty acid beta oxidation and downregulates cholesterol biosynthesis. And in the brain, we were quite interested in the brain. We found there was an increase or upregulation of synaptogenesis and a downregulation of inflammation, but an upregulation of anti-inflammatory genes. Okay? So we were quite interested in this, and we wanted to see how this actually translated into the mice, into a physiological response. I mean, this is just gene expression. So we did this experiment where the mice were put in a water maze, and there was a video, and we could just measure and monitor the movement of the mice. And there is a little platform here. And what was computed was the time taken for the mice to reach the platform. You can't quite see the platform here. It's kind of transparent. So we noticed that the mice that were on phenolics took a significantly shorter time to reach the platform. If you look at the speed at which they were swimming, there was not much difference, no significant difference in the velocity in swimming, but there was a significant decrease in the distance, meaning that these mice remembered and they took the shortcut. Okay, so the enhancement of cognitive function here. Okay, and in this experiment, the same mice, they were put on a rotor rod, and the rotor rod was spun faster and faster just to see the average time that this mice would spend on the rod. And again, there was a significant difference. Those that were on all palm phenolics could stay on the rod faster, suggesting that there was improved motor function. Okay. Now, more recently, together with MIT, we've also found that all palm phenolics, and therefore palm oil meal effluent as well, is a very rich source of shikimic acid. Now, this is a highly valuable substrate for synthesizing Tamiflu, and it's probably the most sought-after drug in the world, especially when you have a pandemic like we did during the H1N1 pandemic. The current source is mainly from star anise, and this from China, and this might be an opportunity for Malaysia to break China's monopoly on shikimic acid, okay? Because, I mean, if you've got 45 million tons of palm oil meal effluent, there's a lot of shikimic acid you can get from there, okay? Now, what's our dilemma? Everything seems to work. It's almost like a snake oil, and we don't want a snake oil, right? So we agree. So we are very mindful of the fact that whatever we do has to be substantiated by science, and we have to strategize so that it doesn't look like we are selling snake oil, okay? We have published in different journals, okay? And we have filed several patents. And we keep reminding ourselves that we must stay focused. So I think there are people who might be interested in this product, and at least for a first step for commercialization, we would like to look at the effect of all palm phenolics on macular degeneration, neuroprotection, diabetes, and probably the fourth would be heart protection. So rather than all the rest, because it's just too much to do, but this is what we're going to do, stay focused on these three, for the time being at least, okay? And what's the way forward? We need to move on and have toxicology studies that will give us GRAS status. We already have self-affirmed GRAS, so it's one more step, and that's going to be carried out shortly. We need the human intervention trials, okay? One on wellness and another one on diabetes. Later this afternoon, you will hear Dr. Fairuz's presentation on the phase 1 studies we've done on all palm phenolics, which he carried out. And the next step is, of course, go on to diabetes, and Julia has got some plans for this. And finally, we would like to commercialize it so that we can make this available to more people. And Prof. Sinsky will talk more on that later in the afternoon. But as far as we are concerned, we believe that there's huge potential for oil palm phenolics in several industries: functional food, nutraceutical, pharmaceutical, cosmeceutical. Okay? We have a winner because we have abundant raw materials, simple solvent process, inexpensive processing costs, and Prof. Sinsky will talk to you about this later. And what is the impact? Definite health impact. If we can translate our studies, of course, into humans, and we have to do that. Obvious economic impacts, because you're starting with basically a material that's being thrown away, and environmental effect as well. So it's basically green technology, golden opportunities from this green technology, or green opportunities from our golden crop. And I'd like to invite you all to PIPOC as well. The theme of PIPOC this year for MPOB is green opportunities from the golden crop. And with that, I'd like to thank you, and also to acknowledge the tremendous support of my Director General, Dr. Chu Yun Mei, as well as the rest of MPOB management. Thank you so much.